Now in this video we want to be thinking about the process of absorption, and the point of anatomy of particular interest for this purpose is the small intestine. So here's the overall diagram of the gastrointestinal tract, and the small intestine is going to stretch from the pyloric sphincter at the end of the stomach all the way through to the ileocecal valve, where the ileum joins onto the joins into the cecum. Now, as we see from this diagram, the small intestine is in three components. We have the duodenum, the jejunum, and we have finally the ileum as the distal part. So the duodenum is proximal, the jejunum is in the middle part, medial, and the ileum is distal, parts of the small intestine. And it's called small because it's more narrow. As we see, the lumen is more narrow than in the large intestine. Now, the length of the small intestine varies a fair bit between individuals. Studies have shown it's between 3 and 8.5 metres in length, with the average being round about 5 metres in length. Now, you may well see longer figures than this quoted in the textbooks, and that's because the textbooks are based on old data which is taken from post-mortem studies. But the 5 metre average is the length of the small intestine as measured during surgical procedures when the patient is alive, because at death the smooth muscle in the small intestine will relax and the small intestine becomes longer if we measure it on the bench. But in all probability your small intestine is about 5 metres in length. Now, what we want to do now is look at these structures here. The small intestine, the circular folds, the villi and the microvilli, because it's this anatomy that gives the small intestine a remarkably large internal surface area, the surface area between the mucosa and the submucosa of the small intestine and the lumen of the small intestine through which absorption takes place. This very large surface area is facilitated by this particularly interesting arrangement of anatomical and histological structures. So first of all, let's think about the small intestine. So here's the length of the small intestine, just here, and it's got a muscular wall. We could draw a muscular wall in there. So there's the muscular wall of the small intestine. Right, and inside, as we know, there's a mucosa, and under the mucosa there is a submucosa. Now, the submucosa is arranged in folds like this. So it's folded on the inside. These folds that project into the lumen of the small intestine. And I think if you look at those already, you can see that we have a much larger surface area exposed between the lumen and the submucosa here. So this is the submucosa. This black line is actually the mucosa. This is the surface mucous membrane of the small intestine. And we see that these circular folds are greatly increasing the surface area. This would be the submucosa just here in this area here. In fact, we could just colour that in to clarify it. So this area here is the submucosa. And the submucosa is so important because it contains the blood vessels which are going to collect the absorbed nutrients. And the venous branches are going to go to the liver via the hepatic portal vein. The lymphatic branches are going to go into the lymphatic system to be drained back into the blood separately. So there we have the submucosa. So what we've drawn here are the circular folds. These are circular folds. So that's one circular fold, that's another circular fold there. So large circular folds of mucosa project into the lumen of the small intestine. This is the lumen in the middle here, the gap down the middle. Now, these are not present in the first part of the duodenum, and they disappear in the distal ileum. But the rest of the small intestine has these circular folds. And they're visible macroscopically. They're up to 8 millimetres deep. And one large fold actually alternates with a smaller fold. So it's a fairly precise arrangement of these circular folds. And we can actually look at this anatomy in some more detail. So let's imagine these are the circular folds here. These are the circular folds, a little bit larger this time, blown up a bit. So they're the circular folds in the lumen of the small intestine. Now, as you probably know, and as we've mentioned, the walls of the small intestine are lined with muscle. So just here, there's going to be a layer of circular muscle. So this muscle is circular in nature. The muscle fibres running around about the diameter of the gastrointestinal tract. Then on the outside of those, there's a layer of longitudinal muscle fibres that run longitudinally. Of course, we need both circular and longitudinal to facilitate the process of peristalsis. And then finally on the outside here, there's a layer of serosa, the outside tissue of the gastrointestinal tract. And at the level of the small intestine, that is going to consist of the visceral peritoneum, the visceral peritoneal membrane. And this fairly large area here, this submucosa, that's going to contain many blood vessels. It's going to contain lymphatics. There's going to be lymphatic vessels here as well. There's going to be lymphatic vessels draining away into larger lymphatic vessels in this submucosal layer. So we've got the small intestine and the circular folds, but the next layer of complexity is the villi. So what are these villi? Well, if you've got good eyes, the villi are just visible to the human eye, and the villi are present on the circular folds like this. So these would be the villi, not drawn to scale here, much smaller than this. The villi would be in this position here. So these will be villi lining the circular folds. So the actual mucous membrane itself is enfolded in these villi. It goes up and it goes down with the villi. And as we've said, these villi are just visible to the human eye. And the villi are actually highly vascular projections of the mucosal surface. And they cover the entire surface of the intestinal mucosa. The small intestine is completely lined with these villi. And what they do is we can see that the surface area has already increased with the circular folds, while the villi, they increase the surface area even more, and in fact the villi alone increase the surface area by times 7. So the surface area is 7 times greater because of the presence of these villi on the circular folds. And there's about 10 to 40 of these villi per millimetre, per square millimetre. So if we take a square millimetre surface area, 10 to 40 villi there. When I said these were visible to the eye, they're probably not visible to mine. If you've got young fit eyes, I'm sure you could probably just see them, but they are quite small and close together. So 10 to 40 per cubic millimetre. And height-wise, they're about 0.5 to 1 millimetre in height. So the scale here is, if that's a good sized villi there, that line from there to there would be 1 millimetre. So next we can look at a villi in a villus in more detail. One villus, several villi. So here we see the villus here. That's one there. Like that. So what we've drawn there in larger magnification is one of those. So we're sort of zoomed into that level now. So we've zoomed in from the circular folds to the villi on the circular folds. Now we've zoomed into one villus. Now between individual villi, there's actually an indented area like this, intestinal crypts. There, like that. Then there'll be another villus there, like that. Then there'll be another intestinal crypt there, and another villus. So that's the kind of arrangement that we see in the villi. Now, in the centre of each villus, we're going to have a lymphatic. It's called a lacteal, so there's a central lacteal in the centre of each villus, and the smaller lymphatic vessels are going to drain into that. And then these are going to connect to larger lymphatic vessels further down in the submucosa, with the lymph eventually draining away into the larger lymphatic vessels. And this is important because the fat-soluble materials are absorbed into the lymphatic system, not into the blood vascular system. And then we're going to have blood vessels here, so there's going to be arterial vessels, small branches going off here like this, and there's going to be an arterial vessel going up, say, this side of the villi, and that's going to send out blood vessels across the individual villus like this. That's going to go into venous vessels, which are going to drain again into, as we'd expect, into larger venous vessels. So we have the lymphatic supply, we have the blood supply going in like this. We have the venous blood draining out, and we have the blind-ended lymphatic capillaries collecting lymphatic fluid and fat-soluble material. Now this, of course, is the lumen of the small intestine here. So water-soluble products of digestion are going to go into the blood supply. The monosaccharides and the amino acids, for example, are going to go into the blood supply. And we can see that they're all going to be drained. All these nutrients are going to be drained via this vessel here, one of the venous vessels. And all these vessels are going to join up. They're going to form the hepatic portal vein, and they're going to take this blood directly to the liver. But fat-soluble material is going to be absorbed into the lymphatic lacteal. And again, that's going to drain away into progressively larger lymphatic vessels. So we've got the infolding of the circular folds, the infolding of the villi, and we've seen the infolding of the villi here. But there's another particularly fascinating area of infolding as well, because the surface of each villus is itself lined with numerous microvilli. So the surface area of this is also greatly enhanced with these microvilli. So we see all these layers of infolding. So the surface area of a villus is greatly enhanced by the presence of the microvilli on its surface. Now, just before we go and look at these microvilli in more detail, let's just use this diagram from the physiology notes books to summarise where we're at. So first of all, we noted that the intestine itself had circular folds. Then we noticed that this is all one circular fold here. That's all one circular fold. We noticed that that was lined with the villi. Then we looked at the individual villi, and we now notice that the individual villi are themselves covered with smaller microvilli, and these microvilli are generated by the enterocytes. And the enterocytes are the most common form of cell lining a villus. So each villus, of course, is going to be lined with, this is the diagram we had of the villi. Each one is going to be lined with individual cells like this. Again, not drawn to scale, but they're going to be lined with individual cells like this, each with their own nucleus, of course, lining the villi, forming the mucous membrane. So there's actually a single layer of epithelial cells covering the intestinal villi. And the most common sort of cell lining the villi are the enterocytes. But there's also other cells. For example, there's goblet cells to produce mucus. And actually, just before we go on, I think I'll just tell you that in these intestinal crypts, so we have these intestinal crypts here. So these are the intestinal crypts. Now, the stem cells to produce the goblet cells and to produce the enterocytes are actually in these crypts, intestinal crypts, what used to be called the crypts of Lieberkühn in the old days. And these are the germ cells. These are mitotically dividing, because it's fairly well known that the gastrointestinal tract is replacing its surface very frequently. And these individual enterocytes actually only live for about 5 days. They're quite short-lived cells. That's why in clinical practice, when we give patients chemotherapy, they often have quite severe gastrointestinal side effects, because that's inhibiting the mitotic activity and preventing this replenishment. So what actually happens is the cells will divide down here and they'll move up onto this mucosa lining the villi, but as they get older they actually physically move up here like this. so a cell down here is going to be maybe 12-24 hours old, but a cell up here is going to be, say, 4 days old. And then eventually the cells get to the top here, up there, and at that stage the cells commit suicide, this process of apoptosis, and they just flake off into the lumen. So this layer is constantly being replaced, and there's this flow of cells from the base up to the apex of the villi, constantly renewing the mucosa of the lumen of the small intestine. Quite amazing. Generation of cells, migration of cells, and then when they've done their job, they just know they've done their job and they commit apoptosis, and that's them. They're replaced by younger cells. But interesting to note, if you look at a villus under the microscope, they're the younger cells there, they're the older cells up at the top. But as we say, only about 5 days lifespan for these enterocytes and goblet cells. So an ongoing process of mitosis from stem cells, migration and sloughing off. The lining of your small intestine is always young. Now, these enterocytes are particularly amazing cells, so let's look at one in more detail. So we've said there's a single layer of epithelium covers the intestinal villi, covers the villi. And these are enterocytes and goblet cells from a common stem cell ancestor in the intestinal crypts. Now, the enterocytes, they're basically column-shaped cells. Nothing unusual about that. They're columnar cells. They each have their own nucleus, of course. And they sit on a basement membrane, as you would expect. And these enterocytes are so important because these are the absorptive cells. This is where absorption takes place from the lumen into the submucosa, where the blood vessels collect the absorbed nutrients. They're actually relatively big cells. They're about 20 micrometers in height. But the fascinating thing about them is the surface of these, as we've said, is folded into microvilli, like this. So this is the enterocyte with the microvilli on the surface. So just to go back to our previous diagram, when we drew the microvilli here on the surface of a villus, these microvilli are actually projections of these enterocytes here. So the microvilli are actually coming from here like this. Actually projections of the enterocytes, and here we see it in greater magnification. Now, amazingly, there's up to 3,000 microvilli per cell. So 2,500, 3,000 of these microvilli on the surface of each enterocyte. It's an amazing level of complexity. Now, we've said these cells are about 20 micrometers tall, but the microvilli themselves, they're only about 1 micrometer. That's 1,000th of a millimeter in height, like that. And this arrangement of microvilli is form what is called the brush border. The brush border. Now, the brush border is a term which was developed during the days of light microscopes. So when you looked at this with several hundred times magnification, you could see it was like a bit like the surface of a brush. You could see it was so many villi there like that. It looked a bit like a brush. So they called it a brush border. This ill-defined border. So this is the brush border. Now, of course, now we live in the days of electron microscopes. We can see these in exquisite detail. So we now know that the brush border is composed of 2,000 to 3,000 individual microvilli on the surface of each enterocyte. Such is the advantage of looking through the electron microscope. Now, if we look at one of these microvilli now in greater magnification, so that's a microvilli there. And of course, this is the surface of a cell. So the surface of cells are made up of phospholipid bilayers. So this is the phospholipid bilayer. Here, we've now blown up one of these microvilli. And the thing about phospholipid bilayers is that there are proteinaceous inclusions that can float on this phospholipid bilayer. So floating in this phospholipid bilayer, attached to the bilayer, there are various proteins floating. This is called the fluid mosaic model of the cell membrane. It's like a fluid, and proteins can float in this membrane, this phospholipid membrane, lining the surface of the microvilli. In other words, lining the surface of the enterocyte. So again, what this means is here is inside the cell, because that's the blow up of, say, here, and here outside, this is the lumen of the small intestine, with the products of digestion here. And this is so important because we know we're talking about absorption. We have this large surface area, but this is also where the final phase of digestion occurs via the action of these membranes, or these membrane-bound proteins. And some of these membrane-bound proteins are digestive enzymes. And as nutrients pass through this membrane here, they're going to be acted on by these digestive enzymes, completing the digestive process. So these enzymes, for example, will degrade disaccharides and oligoproteins, small proteins. Disaccharides will be degraded into monosaccharides, the single sugar units. Oligoproteins, short proteins, very short chains of amino acids, will be degraded into individual amino acids. So even though in the lumen here there can be disaccharides and small chains of amino acids, once we actually get into the cell, thanks to these digestive enzyme proteins situated in the plasma membrane, in the surface of the microvilli, what we get here inside is only amino acids, single amino acids, and only single sugars, the monosaccharide sugars. So just to contextualize this against this diagram, these enzymes here are actually located around here in the surface membrane of the microvilli, these final digestive enzymes. So integral membrane-bound proteins complete the process of digestion at the brush border, which we now know to be an arrangement of microvilli. And if we think about the enterocytes, they're even more amazing than this. Most books draw enterocytes like I've drawn it here. So most books, when they're telling you about these enterocytes, will draw them like this. They'll have a nice straight line there. They'll have the microvilli there. They'll have the next one next door, like this. And they'll rightly point out that the junction between two cells is a tight junction. It's a tight junction. And these tight junctions here are important to prevent non-selective diffusion. So the cell is choosing what's going in. Bits and bobs can't nip in through there inadvertently like that. And this is absolutely true. But of course, having said this, the gut lining in young children up to the age of a few months of age, certainly say up to 6 months of age, the lining can actually absorb ready formed proteins. So for the first few months of life, larger proteins can actually get through this membrane somehow, and they can get into the blood. So for example, if we feed a child cow's milk, that's going to contain bovine albumin. So one of the proteins in cow's milk is bovine albumin. And that means in a child, if we feed a child cow's milk, we can actually get cow proteins actually in the child's circulatory system. Whereas if as adults we drink milk or eat cheese, that's all going to be broken down into its individual components before it's absorbed. But children can actually absorb it as whole proteins. And this is why we don't want to give young children cow's milk, probably for the first year of life. Guidelines might vary, but certainly for the first few months, certainly for the first 6 months of life, and I would probably say up to a year of life, a year of age, to be on the safe side. Because we don't want these proteins getting into the blood. Because these proteins, of course, are not human proteins. They're cow proteins. And that means that these will act as an antigen. These will be an antigen. And an antigen, of course, will cause the formation of an antibody, which is an immune protein. So we'll end up with immune proteins to cow protein. And one of the reasons that may well matter is that in the beta cells of the islets of Langerhans, it seems that there are probably proteins which are a similar shape to cow protein. This isn't completely understood. But if we have a beta cell here, this is a beta cell, and on the surface of the beta cell, and of course the beta cell produces insulin. This is absolutely vital for the production of insulin. This beta cell, which produces insulin. It seems that these antibodies can mistake proteins on the surface of the beta cell for cow proteins. And that means that these antibodies to the bovine albumin can actually attack the beta cells and can potentially kill them, immunologically destroy the beta cells. And if we immunologically destroy the beta cells, then that patient unfortunately is going to get type 1 diabetes mellitus. So this is theorised. It does make perfect sense, and practically we certainly don't want to be giving cow's milk to young children, because this membrane is not as tight as it is in later childhood and in adults. But getting away from this digression, I said that most textbooks draw this as a tight junction, and it's true, but they tend to draw it straight like that. This is even more complicated. So what you actually have, if you imagine that's they're the villi there, microvilli, on top of a enterocyte, and they're the microvilli on top of the enterocyte next door. They're like that. Now the membranes between these two cells are actually infolded like this. So The membranes are actually infolded. You see, that's giving you a much tighter junction. It's just amazing, isn't it? So that one will go off and form this cell, again, infolded junction, next to the next enterocyte. And this membrane here would fall back for this enterocyte, again, infolded junction. So we see that the membranes are even tighter than most textbooks draw because they're infolded. And as well as that, there's proteinaceous desmosomes connecting them together like this, making a really tight junction. So in adults and older children, certainly after the age of a year, these junctions are going to be tight and things can't nip in. They can't nip in unexpectedly because of these tight junctions. Everything's got to go through here and therefore be digested and regulated. Those products are going to go through the cell there and into the submucosa. So we've got the microvilli. There's interesting physiology about those microvilli related to pathophysiology. We've got the villi themselves with the microvilli on the surface. We've got the circular folds with the villi on the surface, and then we've got the 5 meters length of the small intestine itself. And it's estimated that this gives an internal surface area over which absorption can take place of 250 square meters. Quite amazing. I mean, the lungs are probably about 100 square meters in internal surface area inside the alveoli, but inside the gastrointestinal tract we have this 250 square meters of internal surface area. So again, just to clarify this final stage that's going on here, this is the diagram from the physiology notes again, that we can just maybe use as a bit of a summary. So what we have here, what I've actually drawn here is 3 enterocytes. In actual fact, each of these processes would take place through each enterocyte. And the key thing here is this surface membrane of the enterocyte here, where all this clever digestive and absorptive physiology goes on. The external membrane of the individual enterocyte thrown into these folds will actually be broken down as they go through the membrane here into the individual amino acids. Disaccharides such as maltose again will be broken down by enzymes in the surface membrane here into monosaccharides, and fatty acids and glycerol. Oh, that's another clever thing these cells do. They package fatty acids and glycerol in the enterocytes into the chylomicrons, these small fat balls that go off into the lymphatic system and then into the blood. So as I've said, this process is going on in a single cell. So amino acids will go through one cell, sugars will go through one cell, and fatty acids will as well. But I've just drawn it as 3 separate cells just to clarify that process. The final stage of digestion in the brush border. So the brush border is from there to there. That is the brush border, which we now know are these microvilli.